Good morning. Today we are going to learn about anti-metabolite drugs used in treatment of cancer. Anti-metabolites, drugs used in treatment of cancer. So anti-metabolites means uh, drugs which prevent metabolic pathways. If you can remember, the sulfonamide drugs are also one of the examples of anti-metabolites. But here we are going to specifically learn about drugs used in treatment of cancer. That's why finally, they're going to inhibit DNA and RNA. So they're met. So these drugs, from the name anti means opposite, they block the actions of metabolites. They block the um, actions of metabolites. So antimetabolites are chemicals that inhibits use of metabolites. The metabolites means food materials are building blocks of our body. That is guanine, adenine, cytosine, thiamine. So these are the building blocks which we were talking. Now, let us learn some of the important properties of these antimetabolites. Some of the important properties of these antimetabolites. So what are those? You see, their antimetabolites are similar in structure to our metabolites, which I was talking about. Let me go for next slide. You can see it. Hmm. So for example, you can see it here, folic acid. So these are the folic acid, which is our natural metabolite, which is produced in our body. Now, there is a drug similar to it. That's what we were telling. These drugs have structural similarities. So they look like similar. For example, uracil is a natural metabolite. Now, the drug here is 5-fluorouracil. 5-fluorouracil. Here there is actually hydrogen, and that hydrogen has been replaced by fluoride. That is only the dissimilarities between these. Remaining everything is similar. In the same way, folic acid, you can see it. The structures are same. This pteridine molecule, this is PABA molecule, this is glutamic molecule, glutamic acid structure. In the same way, folic acid also, you can say this is pteridine molecule, this is PABA molecule up to here, this is PABA molecule, here also this is PABA molecule up to here, then this is glutamic acid. What are the differences now? Now you can see it here. The differences here is there is amine group at 4th position, and here there is keto group in folic acid. At this nitrogen, you see there is hydrogen, here there is methyl group. So these drugs have structural similarity with that of our metabolites. You can see structural similarities between the biological active compounds and antimetabolites. Now, this is natural metabolite, hypoxanthine, and here instead of keto group or oxo group, there is thio group. Sulfur group is attached. It's similar to that of oxygen, double bond. Then it is called as mercaptopurine. Otherwise, it is called as hypoxanthines. Hypoxanthine. Now, here there is guanine molecule, natural one, and thioguanine. Thioguanine. That's what we want to tell. These drugs, which are anti-cancer drugs, anti-metabolites, they have structural similarity. They have structural similarity. So we can see that properties which we were telling, you see. So they have similar structure to molecule as we discussed till now. Molecules that are used in the synthesis of nucleic acid synthesis. Whatever we have seen, folic acid, uracil, guanine, these molecules purines, they are used in synthesis of DNA molecules and RNA molecule in our body. So this DNA and RNA replication, if it becomes more, you get cancer. So because of structural similarities, few functional groups are different. They block normal functions. So there is no DNA RNA synthesis. So how they block? They block by inhibiting enzymes. When they block enzymes, how they are blocking? By competitive inhibition. So when there is competition, the concentration plays very important role. So they are competitive blockers. And these drugs are mainly acting at the S phase in the cell cycle. If you can remember, G phase, G naught phase, M phase, S phase, like that. S phase is the one where there is synthesis of DNA and RNA. RNA. So they block the synthesis. S means in cell cycle, S means synthesis. They block the S phase of where there is DNA and RNA is formed. So they are cell cycle specific drugs. Now, because of structural similarity and they're blocking the metabolites which are present in our body and necessary for the synthesis of DNA and RNA, they're very toxic to us. So there are some processes which we also require regularly for our hair growth, skin growth, GIT lining. It's completely changed every day. WBC cells, the lifespan sometimes is less than 1 hour, RBC 180 minutes. So in all this process, this replication, transcription, translation plays very important role. So those all inhibited. So they're very toxic to our body. Now, what is the beauty of these drugs is they are very well used in multiple cancers, especially leukemia, especially leukemia, lymphomas, head and neck, breast cancer, pancreatic ovarian cancer, and gastrointestinal cancers. So these are some of the properties of these drugs which are acting as anti-metabolites. Now, we have seen this slide. Because of structural similarity, our body, the cancerous cell, especially cancerous cells, and also we told it is competitive mechanism. When you are giving in competitive mechanism, we give drug in more concentration than natural. So the cancer cell will accept uracil. So will accept 5-fluorouracil instead of uracil. A cancer cell will accept methotrexate instead of folic acid. It will accept mercaptopurine instead of hypoxanthine and thioguanine instead of guanine molecule. So this is an. So let us learn now classification of drugs. Here they're classified into 3 types: folic acid antagonist, then pyrimidine antagonist, and purine antagonist. They are classified into 3 types. Based upon their structure, you can classify, or based upon the mechanism or the metabolite which is blocking, you can classify them into 3 types. What is that? Folic acid analogs are derivatives. Examples of folic acid analogs are methotrexate, widely used drugs, especially for leukemia. Then prolitrexate, pemetrexed. These are the drugs which are widely used for under folic acid antagonist. Antagonist, you can tell folic acid derivatives you can call, or folic acid analogs you can tell. All of them are synonyms. Then pyrimidine derivatives. Naturally, pyrimidine derivatives are uracil, cytosine, thiamine. There is no thiamine derivatives, especially used in cancer. Thiamine derivatives are not widely used in cancer. Now, uracil derivatives are uracil analogs. The spelling you can write this or anything. Both are same, correct. 5-fluorouracil, trifluorouridine, capecitabine. Capecitabine as becoming nowadays widely used drug. We will see it. Capecitabine. Cytosine analogs. Cytarabine, gemcitabine. Gemcitabine. Purine derivatives are analogs. Mercaptopurine, thioguanine. So this is the classification. How did we classify them? Based upon the mechanism of action, that is the metabolite or enzymes it is blocking, or by the structural similarities. Structural similarity, the same classification even for pharmacology or medicinal chemistry. Now, let us go now. These drugs, as I was telling you, see, cell cycle specific. So this is the cell cycle which we were discussing. So what does that? M phase, where mitosis process is occurring. Then from M phase, it can go to G0 phase, that is resting phase. G means gap phase. Then R, it can go to G1 phase. G1 phase. Now at G1 phase, what happens is that all the hormones or enzymes, all the enzymes necessary for the synthesis of DNA and RNA are produced here. So are produced, and then it moves to S phase. S phase is the phase where DNA and RNA are synthesized. So now our drugs, these anti-metabolites which we were speaking, will block here. Then the cycle will move to G2 phase. G means, I told you, gap. Second resting phase. Here it prepares all the enzymes necessary for mitosis process, and it goes to M phase. So every day, for example, WBC cell, this cycle is very, very fast. Suppose if it is nerve cell, only once it occurs, nerve cell will usually go to G0 phase. After developing, there is no further development. Now, all the drugs which we are speaking, anti-metabolites, will fall under S phase of the cell cycle. That's why we told these drugs are cell cycle specific drugs. Cell cycle specific drugs which we were discussing. Now, let us see overview of these metabolites. See how we told the folic acid. In the body, we are telling just rough. We are not going in detail that we studied in biochemistry. We are only seeing the ones which widely used. See now here, see folic acid, which is synthesized in our body. No, it is not synthesized in our body, but bacteria and everyone is synthesizing, and we take externally. So folic acid, in turn, will by metabolic pathways will help to produce purines and pyrimidines. Will help to produce purine and pyrimidine. Folic acid as such will not involve in synthesis of purines and pyrimidines. Its derivatives, we will see in next coming class how it acts. So purines and pyrimidines are formed. That means DNA molecules like ATP, that is deoxyadenosine triphosphate, thymidine phosphate, guanine, GMP, like that. All these will form. Then they are converted to nucleosides, that is base, plus sugar, then it combines with phosphate molecule, which is converted into base sugar phosphate. Then they are the components of DNA. From DNA to RNA are produced. From RNA, you are getting proteins. DNA to RNA is transcription. DNA to DNA is replication process. Then from RNA, so you can get the proteins can convert to enzymes, like this. So this is how overall metabolites, how these important metabolites acts in our body. They help in production of DNA and RNA and enzymes. So let us learn. These are the references which you can go and read, and especially from Foyer's Principles of Medicinal Chemistry, Chapter 7, 5th edition. Wilson and Giswold, Chapter 10. So I have given even the page numbers. You can just go and open that page and read. And one more important book is Graham Patrick textbook, An Introduction to Medicinal Chemistry, Chapter 21. You can read. Now, let us see some of the questions, what we studied till now on this topic. Let us see some of the questions so that you can capture whether you got correct or not. You can assess which of the following is anti-metabolite drugs: mercaptoguanine, cytarabine, fluorouracil, azathioprine. And fifth option is all the above. So guess the answer. Guess the answer, please. So it is all the above is the correct answer. So why? Because you see, mercaptopurine is purine derivative. Cytarabine is cytosine derivative. Uracil derivative. Azathioprine, it's also purine derivative. So all these drugs are anti-metabolites. So which option is correct? All the above is correct. So let us see next question. All the above is correct. So now please go through this question. At which phase of the cell cycle do anti-metabolites act? At which phase it acts? S phase, G1 phase, M phase, G0 phase. Guess the answer, please. It is the S phase, which is blocking, because these drugs have structural similarity. So cancer cell will take these drugs instead of anti-metabolites because their concentration is high and they block the synthesis of DNA and RNA. So the S phase is the phase of the cell cycle where DNA and RNA are synthesized. That it is blocking. So they block at S phase. Next question. We are almost finishing the class. Please try to concentrate. Which of the following drugs are structural similarity to folic acid? Folic acid antagonist. You can check the drug from this one. Mercaptopurine is from the name itself. You can see this is purine derivative. Fluorouracil is a uracil derivative. Capecitabine is cytosine derivative. Methotrexate is the drug which is used to, which has the structural similarity with this drug. And you can also see some of the structure which we placed. So this is the structural similarity between folic acid. I kept wrong. This is the answer. Methotrexate is the wrong. Methotrexate is the correct answer. Next, which of the following drugs has structural similarity to uracil? Uracil drug. Capecitabine. capecitabine is the drug which have structural similarity to uracil. So this is the lecture. If you need any clarification, please do comment in the comment section. I will clarify. Thank you very much. Please subscribe to my channel. Thank you.